Okay, thank you very much for the introduction. I'm super excited to be here and to be part of this webinar series. And as many of you probably, I already listened to many really good seminars, so I hope I can live up to that today. I would like to start a little bit about how plants interact with microbes. I focus on below ground introduction, so I'm going to be talking about rhizosphere microbiomes or root-associated microbes. And my main question as a plant biologist is how plants interact with this microbial community that is present in the soil and how different types of plants can shape this type of interaction in different environments, for example. So very broadly, plants interact with their environment via chemical cues a lot. For example, they attract pollinators via scent and via color of their flowers, for example. Or as you can see here on the bottom left, they make specialized, or they have a really a very complex crosstalk with different kinds of microbes that live in the soil, in this case rhizobacteria, to initiate a symbiosis. And once the symbiosis takes place, they form these very nice interactive structures on roots, which are termed nodules. Also, of course, plants have to defend against pathogens. Here we can see herbivores or ants that are cutting off parts of leaves of a plant. And also there we have a lot of metabolic crosstalk going on. There are a lot of tritrophic interactions explained, where the plant not only informs neighboring plants about the presence of herbivores or pathogens, but it can also ask for help by attracting predators of, for example, pathogens that it's attacked by. Then I want to focus very briefly on the top right here, which is actually a picture taken by a company. It's called Indigo. It's a US company, a startup that exists since a couple of years. And their idea of, or their business idea basically is to make biological fertilizers, which are based on beneficial partners for plants. And we can see one of their fields here. The left side is untreated and the right side is treated with their formula. And we can see that in specific environments, this boost of plant growth and yield actually at the end is really supported by these beneficial microbes that can be added in. And of course, some of these beneficial plant microbe interactions have been known for a really long time. For example, the rhizobia I was just talking about beforehand, or the mycorrhiza. And for example, also Bacillus subtilis is a really well described beneficial microbe. But there are many more to be investigated, and this is a really big focus of the field of plant microbiome interactions at the moment. So what do we know about how plants interact with this very wide diversity of microbes that is found in the soil? Well, we have a couple of hints that were published a couple of years ago by now. The first showing that diversity of microbes that is found in the soil here on the very top is decreasing. So if we look at the microbes that are associated with the rhizosphere, we don't find all the microbes that have been present in the soil beforehand. It's only a subset. And if we then look at the endosphere, or basically the inside of the root, we can see that some of the microbes that were living on the outside of the root on the rhizoplane can actually immigrate into the root and colonize the root interior, but not all of them. So we have this reduced diversity. Also, we know that the type of microbes that are, or microbiomes that are found associated with plants depend on the soil type, which makes sense because the soil is like the original sphere, or the original environment from where microbes can colonize roots at the end. And here we can see a comparison between like conventional farming and organic farming, and we can see a pretty nice distinction of the microbiomes on this principal component analysis plot. Also, microbiomes are shaped by plant species. So this is a study that was looking at different grass species, and we can see a bunch of different maize cultivars over here that kind of group together, and then we can see the maize ancestor teosinte, which is grouping a little bit outside. Then we see wheat and sorghum, which are more distantly related, and bulk soil as a control. So clearly, not only the environment, such as the soil type, affect the microbiomes found associated with plants, but also the plant itself can shape who is there. So how does the plant do that? There are, again, a couple of hints from literature on what plays into that. The first one I want to point out is that plant immunity does play a role on the type of microbiomes that are found. So here we're looking at a bunch of different plant hormone mutant lines that are devoid in specific parts of signaling, and we can see altered microbiomes associated with these. Then also the plants in different developmental stages have been recorded to have different microbiomes, and the same is true for root exudates. And exudates in that case are the metabolites that are produced by the plant and exported from the roots that are found in the rhizosphere and that are available by microbes to use. So here we can see an exudate profile, a principal component analysis plot again of a Brachypodium distachyi on the model grass, and you can clearly see it shifting over time. So that's a correlation, of course, so we can't say that the altered exudation causes the altered microbiome associated, but it's definitely possible that some of these exported metabolites might actually shape the microbial community that is associated with roots. Also, specific compounds, for example, aromatic organic acids here or triterpenes in the other case, have been shown to influence the microbial community that is associated with plants. So in the first study, it was shown that comparing microbes that are found in bulk soil versus microbes that are associated with plants, that they have different substrate specificities. So the ones associated with plants, they can eat these aromatic organic acids, whereas the ones that are living in bulk soil can process those less well. Also, in this other publication, it was shown that mutant lines that are exuding more or less of these triterpenes have an altered microbiome compared to the wild type. So overall, we have a certain idea on how roots interact with microbes present in the soil, and this is kind of how we picture it. So it's very complex, and I want to just give you a very brief idea of we think how that works. So we know that microbes are sensed by plants via their immune system, which leads to gene expression changes, to changes in metabolism, and some of these metabolites are then exported into the rhizosphere. I depicted a little transporter protein here, but actually for most of the compounds, we don't actually know how they are exported from the roots interior into the rhizosphere, which is one of the points I want to get back to later. So these metabolites that are present in the rhizosphere can then be either nutrients or signaling compounds to microbes that live in the rhizosphere, which of course, again, similar to plants, sense these different metabolites. They react with gene expression changes, metabolic changes. They also in turn produce their own exometabolites that are exported from the interior. Then we of course have microbe-microbe interactions that are going on in the rhizosphere, and some of these microbes can then immigrate and colonize the root interior. So overall, it's a very complex interaction, and although we have kind of a picture on how it works, there are still a lot of open questions that go with that. Now I want to, or in my group that I started recently at the University of Zurich, we focus on 3 aspects of this interaction currently. So the first of which is when plants are sensed by or when microbes are sensed by the plant's immune system, how does that then result in gene expression or metabolic changes at the end? So what changes when the plant sees the different microbes? And I already mentioned the export of metabolites from the roots. This is a very I guess, a kind of an enigmatic topic at the moment. So we know that a lot of these metabolites are exported. They're differentially exported depending on the condition, but we don't know if transported proteins are involved or if this is more of a diffusion type process. And then last, we don't really know too much about the diversity of metabolites that is present in the rhizosphere of different kinds of plants in different environments, which is important to understand what's present for the microbes to interact with at the end. So I want to focus on a couple of aspects that play into that in the next couple of slides. And the first study I want to talk about is one that we performed with a model grass I mentioned before. It's called Brachypodium distachyon, and you can see it living here in a little microcosm. It's a sterile environment. We call it the ecofab for eco-fabrication system. And the roots are sitting here in a growth medium, which is either regular half MS growth medium with all the micronutrients that they need, or a phosphate devoid condition, or a soil extract. And the soil extract we generated by taking a regular greenhouse soil, incubating it with water for a certain amount of time, and then sterile filtering it. So what we had was a mix of metabolites that were present in the soil, but without the actual presence of microbes. And what we did was two things. First, we looked at root morphology and metabolism, but we also conducted this study in 4 different laboratories to test how reproducible our data would be. And the short answer to the second part is that it was actually really reproducible. So if you look at the different shapes and symbols here in this principal component analysis plot, you see that they nicely cluster together. So there might be some variation, you know, like the squares are here and then the round shapes are more here, but overall all the different treatments clustered together, which was really nice to see for us. The more interesting biological aspect that I wanted to point out is that not only do the roots look different in these different environments. So for example, the phosphate devoid roots make these longer root hairs, which is to be expected. And the soil extract grown roots have these super long root hairs, which are much longer than the ones grown in phosphate devoid conditions. So not only do the plants react to the different conditions, but also their profile of metabolites looks really different. So here we're looking at root extracts, so metabolites present in roots, but the same is true for the root exudation metabolites. And a last interesting finding that really surprised me at that point was that when we looked at plants growing in the soil extract medium, we initially wanted to see differential exudation in this environment, but the issue was that the amount of metabolites in the soil extract was so high we could barely see any exudation, just because there was already a lot of metabolites present. But on the other hand, we saw a depletion of about 1/3 of all the metabolites that we looked at in the soil extract. So we can't really say that the plant has been taking up all these metabolites. It could also be that, for example, pH changes led to, for example, a hydrolysis of some of these compounds. So we wouldn't see them in our analysis anymore. But still, to me, it was striking that plants, as we didn't give any other nutrients to these plants in this setting, they had to take up whatever was there in the soil extract, and they are apparently very efficiently doing so. In a second study that I want to highlight quickly, we performed a similar type of experiments in a different setting. So we were not using EcoFab, but we used glass jars. And the question there was, how different would the plants grow, again, Brachypodium in this case, if we alter the physiochemical properties of the substrate? So instead of using actual soil, because we learned before that its actual soil has some challenges that go with it, but also to just better control the experimental conditions, we decided to use different sized glass beads to look at the physical structure of soil. Also sand, because it's a more natural environment to glass, but it's chemically very similar. It's very inert, so it shouldn't really disturb the plant too much. And then we added in clay, and clay is a chemically very specific environment in a way that its surface is charged. So a lot of compounds interact with clay surfaces. And we again looked at root morphology parameters, and we also looked at exudation. And first, I want to point out, we looked at a lot of different root parameters, but what I depict here is the fresh weight of roots in general growing in these different environments. And we can see that with smaller glass beads, the root weight actually decreases, which is mostly because probably the roots have difficulty penetrating the substrate at one point. Same is true for sand, but actually it takes much smaller sand granules to come up with the same effect compared to the glass beads. So those are sizes in millimeters. Sorry. We included a hydroponic control without any beads, which gives the baseline here. And then for clay, we see that the plants are very similar morphology-wise compared to hydroponic controls, or the ones growing in bigger size glass beads or sand. Then coming to the exudation profile, we saw actually pretty similar exudation in the different environments. Here we have the hydroponic controls, and we have the differently sized glass beads. We have differently sized sand. We see some differences between the environments, but the most striking difference here is the plants growing in clay. And to figure out if this is actually something that happens on a metabolic basis by the plant, or if it's caused by substrate, we not only collected the exudates in the substrate the plants were growing, but we were also removing the plants from their substrate, and we were collecting the root exudates again. And that allowed us to tell that the exudation profile of plants is actually really similar. Also, the ones growing in clay, they cluster together with the other plants. So the differential exudation that we see here is actually caused by clay. And when we looked at the differences between plants grown in, for example, hydroponics with the one grown in clay, we found that the one grown in clay showed way less metabolites. So many of these metabolites are bound to the clay granules, presumably. So the next question we asked was if these metabolites that are bound by clay can actually be desorbed or used by microbes, for example. So we took a rhizobacterium that we had in the lab at that point and added that bacterium to different sources of clay. So that's the growth control with the defined medium that we mix together, that is based on compounds that are found in root exudates. And we see, depending on how concentrated of a defined medium we give the bacterium to grow, it grows to different densities. Then we were growing the rhizobacterium in clay that was pre-incubated with this defined medium and then washed, or clay that was not pre-incubated with the defined medium. And also we were growing the rhizobacterium in the supernatant of this washed clay. And the interesting point is the first bar graph here, which shows that rhizobacteria can actually use some of the metabolites that are bound to clay to grow. So that probably means that in a natural environment where we have different components present in soil, such as clay and other types, maybe sand, organic matter, there are probably a lot of different niches depending on the structure of the soil where different microbes live. And some of the exudates are probably directly used by microbes that are associated with roots, but others might first be bound to surfaces such as clay and then later be desorbed by other types of microbes. I just want to briefly mention another topic that we are thinking about, which is the involvement of transporters in this process of exudation. And actually, for time reason, I'm going to skip this slide. If you want to look at this publication, it's really interesting. It shows that a sugar transporter, SDP13, is upregulated when the plant is attacked by pathogens. So it can import again the sugar that is present in the apoplastic space. And that kind of indicates that transporter proteins are regulated by the presence of different microbes in the environment. And actually, a couple of years ago, we showed a similar, we had a similar finding in our lab, which was we were characterizing a transporter in Petunia that was localized in roots in these hypodermal passage cells, which are subcellular or subepidermal cells, sorry. And the transporter is present in these cells, as you can see here, with a gas staining, which is overlaid here with an ink staining that shows the hypodermal passage cells because they are permeable by ink, in contrast to the other root cells. And the presence of this transporter allows for the exudation of stragolactone, which is a plant hormone, but also the attracting compound for this mycorrhizal fungi here that are attracted by the chemical gradient and then penetrate the epidermis and colonize the roots at the end. In a later study, we found that not only is the transporter localized in these cells, it very nicely shows this outer lateral localization here, which one would expect for an efficient export of stragolactone. And the importance of the transporter is shown in two experiments here. First of all, the degree of microization of plants that have or don't have the transporter. So here we're looking at wild-type lines, and here we're looking at the stragolactone biosynthesis mutant line, or the PDR1 mutant line, which is this transporter mutant line. And you can see that both the mutant lines behave relatively similar. So they have a lower degree of microization over time compared to wild type. So basically knocking out the transporter has the same effect as knocking out a biosynthetic gene of the hormone, which is pretty striking, at least in my eyes. We could also show that PDR1 is actually really a stragolactone transporter by expressing it in Arabidopsis as a heterologous system. And if we look at this PDR1 overexpressing lines, and we compare the actual room temperature incubated plants with the cold incubated plants as a control, we can see that the amount of radiolabeled stragolactone that we use increases in the water fraction, which is outside of the root, compared with the amount that we find in the root. So there seems to be an active export of radiolabeled stragolactone from the root into the water. So overall, we know about some of the transporters that are involved in exudation, such as, for example, this Petunia PDR1 that I just mentioned, and we also know about a couple of other ones that are listed here. But many compounds that are found in a root exudate, such as amino acids, nucleotides, sugars, for example, the mode of export, as I mentioned already in my introduction, is still unknown. So this is one of the focus or a focus area of our lab at the moment, to try to understand if more transporter proteins are involved, and if yes, what happens if we differentially regulate the amount of the transporters present. With this, I would like to thank my group, Alex and Sarah, and a lot of my mentors, such as Cyril and Uwe, who were a big and still are a big support in my current research, and collaborators from across Switzerland and abroad. And with this, I'm happy to take questions, and I thank you for your attention. Thank you.
Thank you very much. That's great. Very interesting stuff. And as you said, you've just set up your lab the week before lockdown started last year, so it's been a challenging time for sure. So making some good progress. So I'd invite anyone to ask a question into the chat box that I can pass on. But let me first of all ask about the work you did, that PDR1 work you did in Petunia. So yeah. How did that? I'm intrigued by the process of how and why did you start looking at this in Petunia as opposed to more conventional model systems?
Right, yeah, that's an interesting story, and it actually started way before I joined the lab. So that was my PhD project at the time, and it was started by another graduate student who was there beforehand. And the idea was to try to understand better. It was a lab working on ABC transporters, and it was well known that ABC transporters are important for hormone transport. So at the time, it was known that stragolactones are the compounds that attract mycorrhiza. And it was also known, of course, that, for example, Arabidopsis is not a species that forms these mycorrhizal structures. So it was clear that Arabidopsis would not be the model of choice. And there are a bunch of different groups in Switzerland working on Petunia as a model system. So there is a good community that is established on that plant. There are a bunch of molecular methods that are present, so that was one reason, and the other reason is that it's mycorrhizing well, and there is actually a group working on Petunia mycorrhization in Switzerland. So I wasn't there when the decision was made initially, but I can picture that, like the setup of like the mycorrhization and the Petunia and all was really helpful. And then I know that at the time they screened expression of a bunch of ABC transporters that are often regulated by their own substrate. So they, at that time, they looked at phosphate starvation because those are the conditions when mycorrhization is initiated. And they looked at ABC transporters expressed in roots. And the PD-L1 was one of the candidates that came up.
Okay, so we have a few questions come in now. So question from Sebastian Feilmeier, and he asks, I have a question regarding the clay changing in the in situ exudates. Does the clay modify exudation by the plant, or is rather absorbing it and making it biologically inaccessible?
Yes, so I would argue, looking at these PCA plots, that it's probably the latter, because we were growing plants in these different environments, and when we collect exudates within the substrate, we can see that the clay is really grouping apart from the other conditions. But then if we remove the plants from clay or from whatever substrate, and we collect exudates again in falcons, we see that all of the exudate profiles overlap, which means that the exudation of the plants is initially the same by all the plants in all these different substrates. Hoping, of course, that within the couple of minutes that we remove the plants from the substrate, not much is happening. But yeah, at least what we can see here is that a lot of the compounds are depleted by clay. So I would argue it's probably not the plant metabolism that is changing too much in the clay environment, but it's rather the clay surface that is really altering the metabolites that are present or that are accessible for our LCMS analysis or for microbes to eat, for example.
Okay, so we have a couple of related questions from Kawai Ma and Rusko Hardy, and they ask, they say, in the beginning you mentioned that the exudate profile changes with developmental stages. Does that change have any known adaptive advantage by altering the interactions with the microbiota with the plants at all?
So I'm not aware of a study that actually looked into that. There are a lot of, or there are some correlative studies that some of it I mentioned in the beginning, that were kind of correlating different, for example, genotypes with different exudate profiles or different microbiomes. So we do know that it correlates, that different exudates correlate with different microbiomes. But to really know if that's a causality, we would have to learn much more about substrate specificity of the different microbes. So if the microbes that are associated with later developmental stages, for example, metabolize better the type of exudates that are present there than the ones that are found in earlier developmental stages, for example. So I know that this is a very complex question because it not only depends on the substrate specificity of the different microbes, which requires cultivation and growth of microbes in these different exudates to understand, but there are also a lot of microbe interactions going on, and the microbes don't necessarily do the same metabolic from a point of metabolism if they are alone than if they are growing in a community. So it is a very complex question, but it's true that some of the compounds that are exported definitely have signaling function. Some of them are important to attract beneficials, for example, others to attract rhizobia, for example, which are symbionts. So I would argue that probably, yes, it's definitely a hypothesis that is worth looking at, but I don't think we can answer it at the moment.
Okay, great. Okay, so I think we'll leave it there. Thank you.